All right, guys, we're going to pick up our talk here of evolution by discussing some of the pieces of evidence for evolution. The title on this slide says Evolutionary Theory provides a cohesive explanation for many kinds of observations, and so we're going to look at some of those observations that we can see in the natural world that help to kind of explain this idea of evolution. And so some of the things I'm going to talk about here are listed in front of you. And you can look at many different aspects of living creatures to gain an understanding of how evolution, the idea of evolution, is supported in the things that you see. So we're going to talk about some morphological homologies, some molecular homologies, the biogeography of where some of these animals live or developed, as well as the fossil record as it pertains to how these things kind of work as evidence for evolution. So let's talk first about homologies. The term homology means that an animal has some kind of similarity to another animal that results from a common ancestor. And homologous structures are anatomical resemblances that represent variations on a structural theme present in a common ancestor. And what that means is that certain organisms might share, for instance, the example I'm going to show here is bones, but those bones might have variations in them in size and structure, but they're still the same bone. And when you see variations in size and structure of the same bone, oftentimes it leads to different uses of that bone. So what we see here on the diagram in front of you is the forelimbs of 4 different animals. You see human, cat, whale, and a bat. And all of the different bones that you see in this diagram are actually colored similarly. Okay, all of the bones are colored different colors, but some of the bones share some color. Okay, so for instance, if we look, let me get my pointer here. Okay, so for instance, if we look at the top bone here, I believe this is the humerus. It's colored gray in each one of these animals, and those would all be considered humerus in their respective animal. Now, obviously, each one of those bones is different, but they are the same bone. This is why we call them a homology, because they are the same bone. But as you notice, in each one of these different animals, it might be a different size, it might be a different shape. And the reason for that is because they're used for different things, because these animals all have different structures themselves, and they live in different places. But the idea here is that each of these animals has a humerus. If we go down to the lower bones of the arm, we see this red one is analogous to all of these, not analogous, pardon me, homologous to all of these. They are the same. And then this tan one is the same bone, even though each one of those is kind of a different shape, a different size, they're still the same bone. Okay, and then down at the bottom we see the little fingers in the human, the phalanges, all are homologous in the other animals, but again, they're different sizes, different shapes, because these other animals have different environments they live in and different ways that they interact with those environments, and thus their bones have evolved to these different sizes, even though they're still the same bone. Now, another way of interpreting this or adding to this, I'm going to try to make a picture of it. Oh, bugger. Okay, hold on. Is to think about it this way. If we want to try and connect all of these animals back to like a common ancestor, we don't really know what that common ancestor is, but at some point that common ancestor would have had this gray bone, this one here, okay, and it would have had just one shape to it, okay, and it would have had this red bone and a red bone and a red bone and a red bone, but it would have had just one shape to it. So as we see throughout the course of time, that's what these lines kind of represent, is the course of time. The shape of those bones and the size of those bones has changed to suit a particular animal's environment better, and that's what evolution is trying to tell us, is that these structures like this, even though they are the same, ultimately will be changed to fit the animal's environment and help it survive better in that environment and do what it does. Okay, so this is an example of a morphological homology, a form-fitting homology, if you will, part of the animal's form, part of the animal's body. Another example of a morphological homology is when we can look at the embryos, or I should say the developing kind of organism before it's born, the embryos, we can look at those and we can see similarities. And so what you see in front of you, and they call this comparative embryology, comparing embryos, right? And if we look in front of you, you got a chicken embryo and a human embryo, and it points out specific things on each one of these pictures. And specifically these things called pharyngeal pouches and these things called postanal tails. It's interesting to note that the humans during their development actually have tails. Now that tail eventually becomes eaten away through the process of apoptosis, and we no longer see it. And the same thing with the pharyngeal pouches, that those are things that we just don't see in adult animals. And these are remnants of things that might have needed those particular structures in the past that were related to us. Okay, so when we look at some of these embryological homologies, it reveals some pieces of anatomy that aren't visible in the adult organism because they've either been closed off or chewed away, eaten away by apoptosis. And so we can't literally look at the adult organism and say, okay, this is a thing that was shared long time ago. And some of the activities that we covered the other day will help us get a better understanding of this comparative embryology. One of the things that you will notice, and is kind of connected to this idea of comparative embryology, are looking at vestigial organs, where we look at certain animals and we notice things about those animals that just don't float. Sometimes you can look at a snake and you can there are some breeds of snakes that actually have vestigial legs, which is a throwback to one of their ancestors that at one point did have legs. The snake as we see it now no longer needs that leg, but it's got this little remnant, little chunk that looks like a leg, even though it just kind of doesn't do anything. Okay, and that's what we call a vestigial organ. It's a remnant of a structure that serves some important function to that organism's ancestor. The same thing here. We see a human born with a tail, and sometimes this does happen. Sometimes people are born with the little tail nub. Apoptosis didn't do its proper job and chew away the tail over time, which is what we see happening right here. There's a postanal tail here that becomes shorter and shorter through the embryological development of that creature, of that animal, and then by the time that animal's born, there's no tail on it. Sometimes the mechanisms just don't work right, and we can see over here that some humans are actually born with tails. And so this is supposed to be a piece of evidence that tells you, okay, at some point long ago, our ancestors had need for a tail, and obviously we no longer do, so during our development our bodies eat it away, and then we never see it. So this is some evidence to help us understand that, we had ancestors that at one time needed a tail, but we no longer need it, but sometimes it's still there. And it's definitely there during parts of our development, as you saw with the comparative embryology right here. All right, we can also delve into molecular homologies. And in molecular homologies, it's very similar into looking at the morphological homologies. Looking at the body's form for similarities is very similar to looking at the body's DNA for similarities. Okay, so organisms that tend to share this similar DNA sequences are likely to be more closely related. That's how they're getting this. I think we share like 98 or 99% of our DNA with a chimpanzee. So, which means at some point in the past that we did not evolve from chimpanzees. We shared a common ancestor with chimpanzees. It doesn't necessarily mean that we were chimpanzees and now we're humans. It means that we shared a common ancestor. at some point. So the nature of these similarities may suggest that organisms descended from common ancestors. And these homologies can show divergent points between two different species to show where they started to diverge from one another on the family tree. When we get into chapter 25, we'll start to talk about phylogeny, which is the family trees of life, and those are really pretty interesting to put together. And they can put together family trees based on DNA similarities. So if you look at this diagram off to the right here, it gives you two different DNA sequences. The way you would interpret this is by saying, okay, species 1 has this, or individual 1 has this, individual 2 has this. And so what we have to think to ourselves, okay, this was one species, two individuals from one species, maybe one population at some point in time. And over the course of time, there would have been mutations, there would have been, like it shows here in diagram 2, deletions and insertions of each one of these two genetic lines. And as time moves on, because of these mutations that accumulate in the genome, we start to see these two diverge from one another. Okay, so here they're the same, and then as mutations start to stack up on each one of these genetic lines, they start to diverge from one another, and that's where you start to get two different species. Here's a new species, here's a new species. So it is mutation that drives this speciation, and then evolution, natural selection acts on those mutations in the wild, right? And so as we work our way through these last two parts of this diagram, we can kind of see that there is some remnant of the original DNA sequence, which is highlighted here in yellow, but the stuff that's still in light blue that we see are the new changes to these specific DNA. But what researchers can do is that they can, with their new fancy crazy computers, they can still align all of these previous DNA sequences to tell if two things are related, okay, or to tell if they had some common DNA in ancestors long ago. And so that's what we see here in diagrams 3 and 4, is even though that we do have mutations that stack up and take this species and veer it off this way and this species and veer it off this way, these species still contain some of that original DNA that they shared up here in the first diagram. And the computers, now that even though they're two different species, can recognize the similarities in their DNA and say, hey, yeah, you two are different species, but you were related at one point, because we can see all of the commonalities that you share in your DNA all the way back up to this diagram here. Okay, so that's how we can interpret molecular homologies, to say that ancestors who share, or I should say organisms or species that share a large portion of their DNA, can be said to have had some kind of common ancestor in the past. Okay, and so when we can say that, we can say that even though we had this common ancestor in the past and we both veered off from it, we still shared some ancestor at some point in time way back there, and then we had a bunch of changes that made us go these opposite directions. Those changes are evolution and natural selection. Okay. All right, let's talk about biogeography and convergent evolution. Darwin's observations of biogeography, the geographic distribution of species, formed an important part of his theory of evolution. He noticed that some similar mammals have adapted to similar environments, and in fact they evolved independently from their ancestors. And a really, really good example of this is comparing the sugar glider, which is a marsupial and is found in Australia, with the Eutherian flying squirrel. And Eutherians are, womb-carrying creatures. They're like us. We would be an example of a Eutherian, and a flying squirrel or squirrel is really no different. But what you'll notice about the flying squirrel and the sugar glider is that they share very similar morphologies. They share kind of these webbed feet to hands, and they can glide from tree to tree. And really the reason that they developed this similar morphology or form is not anything to do with sharing DNA so much. They're actually pretty far apart on the DNA family tree. And we know that because one's a marsupial and one is a eutherian. They're pretty far away on the evolutionary family tree. But they developed very similar features because they live in very similar environments. And so this is an example of evolution being driven by the environment, and that when you have an animal that has a similar body type to another animal and they develop in or evolve in very similar environments, then you get very similar adaptations. And we watched a video just the other day about the pocket rock mouse, or rock pocket mouse, whatever the heck it's called. And this mouse developed a specific darkly colored fur trait in 2 different ways. So we see this happen in more than just flying squirrels and sugar gliders, but this is something known as convergent evolution. And convergent evolution, if you can think of the term converge means to come together, right? So if you've got 2 things, they come together. And the way we can understand convergent evolution is we have these 2 creatures that are ultimately going to become flying squirrel and sugar glider. They converge on a similar adaptation. And that similar adaptation, or form in this case, is that webbing between their hands and their feet so that they can glide from place to place. So that's what we would call convergent evolution, where you have 2 kind of unrelated things that converge on this similar body form. And we see it in creatures that live in water too. Here we see 3, 3 distinct different types of species. We see fish, we see birds, and we see mammals. And each of these couldn't be more different. They're all 3 relatively far apart on the evolutionary family tree. They probably don't share a ton of DNA because they are so far apart, but they've all kind of converged on this similar body form. It's fusiform. It looks like a missile. They have very slick coverings, okay? They all kind of have some kind of flipper feature that helps them steer through the water. And the reason for this is not because they share a common ancestor, but because the environments that they happen to live in dictated which features are going to help them survive the best. And so they all kind of converged on this similar body form, okay? So evolution being driven by the environment and random mutations is what it's all about, okay? We can also look at the fossil record. Okay, the fossil record provides insights into the past. You can look back and see the fossils, the bones of other animals, and really that combined with morphological homologies and things of that nature, you can gain insight as to what might have been related to something we see today. And so the quest kind of still, in some cases, continues for the quote-unquote missing link between monkeys or primates and human beings. They have all of these transitional fossils. So we've got humans here, and we've got the most primitive apes here, and they've seen a lot of little stepwise fossils and bone structures all the way up through, but there's always that missing one. And that's why I put down here that scientists have found many of these transitions, but they haven't found them all, okay? And they're always constantly looking for that missing link, because it's never, as far as they can tell, it's never a perfect transition from one species to the next. There's always something you're like, well, this bird-like dinosaur creature looks pretty good, but it's still not a bird. There's something in between here that we haven't found yet. And so, the world's a really big place, and you think about how much it's changed over millions of years. It very well could happen that they never find some of those. Some of those fossils might very well be buried at the bottom of the ocean. Some of them might have been turned into lava. You just don't know. So Darwin did predict that those transition fossils should be seen in the fossil record. You find a lot of them, or scientists do find a lot of them, but they still haven't found every single perfect transition, and that there's various reasons for that. So one of the things that the College Board does, for whatever reason, want to get into a little bit is fossil dating, and how we can date fossils. And so I've provided a couple of different ways that can be done here. You can radio date, or carbon. You guys call it carbon dating, but the technical terminology for it is radiometric dating, where we look at isotopic decay of certain ions for certain items, in certain items. And the stock kind of default mode is that everybody says, oh, I'm going to carbon-14 date this. I'm going to carbon-14 date that. Oh, you found a dinosaur bone, go carbon-14 date it. And that's because that's the one that's probably the most famous. It's the one that we can utilize very easily. But it's also one that you can't actually use for like dinosaur bones. Dinosaur bones are just too old. These different radiometric dating techniques do have a quote-unquote shelf life, or we could call it a half-life for real, where by virtue of the atoms, or the radioactive atoms that you're looking at, only have a certain half-life, so you can only date fossils and things like that you find up to certain time frames. So for instance, I think I read that carbon dating, you can only with certain amount of accuracy date things that are 77 to 100,000 years old with that particular system. Potassium-40 dating, on the other hand, is a way to date things that are much older, in the millions of years, because it uses a different isotope with a different half-life. That being said, potassium-40 dating is not as accurate, but it'll still give you a pretty good estimation as to how old something is when it's in the millions of years old. Okay. You can also look at the age of rock or the earth where the fossil is found, and you can look at geographical data. Okay. So let's just spend a little bit of time on each one of these. Not spend a lot of time, just a little bit of time kind of describing what radiometric dating is, and also geographic dating. Radiometric dating uses a technique where they look for certain types of radioactive isotopes. For instance, everything, like we are all born, things that are organic are born with a certain amount of carbon in them, right? Most of the carbon inside of us is carbon-12, but we do have some carbon-14 in us, and most of the living organisms that we see now have a certain proportion of carbon-14 in us. And what actually happens is that when we die, our body stops incorporating carbon into it, and we have now got this finite amount of carbon-14 in our bodies. And so what carbon-14 does over time is it decays into a different atom. I think it's nitrogen-14 is what it decays into. And so what they look at over time, they can take a sample from this fossil, tear a piece off, grind it up, do some analysis on it, and they can tell what is the ratio of carbon-14 that's still left in the sample to nitrogen-14, which happens after the stuff has decayed, after the organism has died. And they can put dates on this, so based on the half-life of this process of carbon-14 decaying into the nitrogen version of that isotope. And it allows them to get a fairly accurate understanding of how many years have passed by since that thing died. So once this thing dies, the carbon-14 starts to decay over and over again, and that decay happens in a certain amount of years, right? And so based on how much carbon-14 they see left and how much nitrogen-14 is there, they can get a pretty accurate understanding of how many half-lives it has gone through, and then they can tell how old the thing is. Okay, with the geologic data, they can look at these layers of the Earth and they can find a fossil in it, for instance. And these layers of earth are relatively constant across the earth. Okay, so you can see, for instance, they find this skull in this gray layer of earth in Montana. That gray layer of earth is roughly the same age as that gray layer of earth in Texas. And so if they find the same fossil in the same spot, they can, or in the same layer, they can say, okay, this animal was definitively in Montana and Texas at around the same time. And so they can date these layers of earth as well. They can do radiometric dating on the rock as well. And so if they find a fossil in that layer of rock, they can unequivocally say, okay, this snail fossil was in this rock, so I know it's this old. And so that's how they figure out the age of certain fossils based on different layers of the earth that they find them in. Okay? So I hope that is enlightening. I hope it sheds a little bit of light on some of the activities we did the other day. And we are hereby done with Chapter 22. So I'll talk to you guys soon.